Hello everyone. I'd like to introduce to you my idea of transposable element machinery-directed defense modulator system for plant pathogen defense. Now, it's been known for quite a while now that transposons, or jumping genes, exist across multiple different organisms and exist in different fashions as well. Now, the original transposon research was done in maize, and we found out a lot about them. We found out that DNA transposons use a cut and paste technique, where they have this transposase gene encoded within a random location of the genome. Once that is transcribed and translated into transposase, those transposases cut that region out and insert in a different region that is homologous based on specific ITR regions. What's important here is these ITR regions aren't known to be variable necessarily, but what if we could modify them and make them variable and make those regions specific to what we'd want to introduce into a gene of interest? So, for this example, we think about plant receptors for defense perception, and we know that they're highly variable to new and existing pathogenic substrates. In some plants, we can bind a pathogenic substrate, and in others, we see no binding at all, and thus no downstream defense response. So what if we could control the available pathogenic substrate binding domains by switching them out? What if we could make variable regions that are recognizable by the transposable elements for cutting and use that to insert new domains in our existing proteins to try to improve the binding against pathogenic substrates? Additionally, what if we could prepare sequential and specific insertion sites to expand pathogen perception? What if we could use something like this on the right, where we have, for example, a circular plasmid that encodes both partial transposase gene cutting regions as well as the region that contains a specific substrate binding domain and a full-length transposase to make the cutting protein. Altogether, with specific regions that can be recognized by the transposase that also exist in the partial transposase gene cutting region, could we switch and move and add new specific substrate binding domains to our existing plant pathogen perception receptors? These are some interesting questions, and if we could, would we be able to efficiently study and explore how the addition of new regions along the same protein provide extra support and potentially even a greater level of defense against a variety of new and existing pathogens?